Can you still hear us?
Yeah,
We
perfect.
disconnected one screen, but yeah, okay. So please.
Yeah. So good afternoon, everybody. My name is Andrei Belykh. So I'm a new postdoctoral researcher here. As Guido said, I'm working on a project called Mandarin, which aims to evaluate the impact of menopause and sex-related events and hormonal changes to the progression of atrial fibrillation. So that's why I chose this paper. It was published in Nature Communications, as Guido said, last year by the group of authors from California, mostly. It has classical structure. So first introduction. In introduction, authors discuss the importance of diastolic dysfunction using heart failure with preserved ejection fraction as an example. So in particular, they note that heart failure with preserved ejection fraction accounts for half of all cases of heart failure with multiple comorbidities, including diabetes, arterial hypertension, and cardiomyopathies. Also, patients with systolic heart failure present features of diastolic dysfunction. So diastolic dysfunction somehow important contributor to the morbidity of heart failure. However, it's also important to note that heart failure with preserved ejection fraction is distinct from heart failure with reduced ejection fraction from the point of pathogenesis and effective therapeutic treatment. For example, drugs that effective in reduced ejection fraction have no beneficial effects on morbidity and mortality in heart failure with preserved ejection fraction. And consistent epidemiological data demonstrate that women are about twice as likely to develop heart failure with more symptoms burden compared to men, including significantly increased diastolic dysfunction, enhanced left ventricular stiffness, and so on. And also, compared to men, women have some structural and functional differences in heart, including small left ventricular chambers, lower stroke volumes, high systolic and diastolic left ventricular stiffness at a given age, and so on. So based on the mentioned above, authors decided to investigate genetic factors, including sex differences that contribute to traits such as diastolic and metabolic dysfunction, and somehow to examine the relevance of these findings to human disease by interrogating publicly available human data. To test whether female are more susceptible to diastolic dysfunction in heart failure with preserved ejection fraction, they use so-called two-hit model. It's called two-hit because it reproduced two main risk factors of heart failure with preserved ejection fraction. It's obesity, so that's why they use high-fat diet, and arterial hypertension. That's why they use LNAM as an inhibitor of nitric oxide synthase. And animal developed clinical phenotypes, they claim at least at 7 weeks already. So as you can see here, both males and females demonstrate increase in body weight, fat mass, lean mass, white adipose tissue mass, and so on. And on this background, also glucose intolerance observed, and exercise tolerance was also decreased. And as additional factor, the expression level of fatty acid binding protein 3 as an early biomarker of cardiac damage was increased. So somehow they claimed that the model worked. And upon this model, compared with male mice, females exhibited higher levels of EA ratio, EE prime ratio, and lung weight. And in contrast, left ventricular ejection fraction was preserved, so the model is working. And so they claim that females exhibited increased susceptibility to heart failure with preserved ejection fraction. But it's curious that authors note that there was another research, the same model. Their authors found that males more prone to heart failure with preserved ejection fraction. And they explain this difference because of using different genetic background of the males. Somehow here it was C57BL6J, and there it was C57BL6N. But to confirm that this is really related to genetic differences, they choose 30 different strains and measure E prime ratio there. And as you can see here, this ratio somehow reflects dysfunction. The study dysfunction was slowly for me, but significantly increased in females. So based on this data, authors declare that the males somehow more prone to heart failure with preserved ejection fraction. Next, they hypothesized that diastolic dysfunction could be associated with the mitochondrial function of mitochondria. So they analyzed data from previously published study. Unfortunately, they didn't provide reference for this study. It's quite strange. So in this study, they used so-called HMDP. It's a hybrid mouse diversity panel. So it's a cohort of more than 100 strains, genetically diverse, but with high-resolution genetic mapping. So mice from this hybrid mouse diversity panel were subjected to isoproteinol infusion for 21 days. Isoproteinol, it's a non-selective beta agonist. And after performing this, they perform a global transcriptome profiling and revealed genes that significantly correlated with EA ratio. And these genes, pathway in which they're involved, somehow strongly enriched in pathways related to mitochondria, to Krebs cycle, to mitochondrial inner membrane. So this data suggests that alteration in mitochondrial metabolism and bioenergetics linked with diastolic dysfunction. And moreover, correlation showed that in the ventricles of these females, upon isoproteinol-induced cardiomyopathy, mitochondrial DNA content was negatively correlated with the different multiple cardiac traits, such as lung mass, left atrium mass, interventricular septal thickness, ratio of IVS to posterior wall thickness, both in width and as you can see in upper plots, and without outliers. So they conclude that this may suggest that mitochondria contribute to diastolic properties. Next, they measured heart mitochondrial DNA levels among this white panel, and found that mitochondrial DNA content in females upon Western diet, so it's high-fat, high-sucrose diet, significantly lower among all of this white number of strains, significantly lower compared to males. Using also the expression of genes which are related to the mitochondria, both mitochondrial genes themselves or nuclear genes, which code some mitochondrial-related proteins. As you can see here on this volcano plot, so the expression of the genes in females was dramatically lower, and also genes of electron transport chain was dramatically lower expressed, both without normalization and after normalization. And also they used previously published data on human hearts, both failed and healthy hearts, and did the same expression analysis, and found that generally the picture is the same. So mitochondrial genes and electron transport chain genes significantly less expressed in females compared to males. So they suggest that the genes as well as mitochondrial DNA is sex biased, and they are lower in females. Next, to determine whether the sex differences affect mitochondria, and this difference are due to hormonal changes or chromosomal effects. So they performed gonadectomy in absolutely normal conditions, and gonadectomy resulted in reduced mitochondrial DNA content in males and increased mitochondrial DNA content in females. And consistently with this, treatment with hormones somehow abolished this effect. So testosterone increase, normalized, and estrogen decrease. So next, they reproduced this gonadectomy, but also in a two-hit model of heart failure, the preserved ejection fraction. So as you can see here, the general trend and phenotype was preserved, so body weight increase, fat mass increase, and so on. But gonadectomy in males abolished these changes, and decreased the weight gain, decreased lean mass. But in female, it was reverse phenotype. Somehow, gonadectomy in females activate this two-hit model-induced changes. And consistent with the decreased mitochondrial number in males upon gonadectomy, the function of mitochondria was also changed and somehow decreased. As you can see here, they observed reduced activity of state 3U maximal respiration, and also reduced electron flow in complex 1, 2, 4, and 5. And in females, consistent with the increased mitochondrial DNA content, activity of these complexes was increased. So they declare that hormonal regulation is crucial for heart's bioenergetic. And the next logical step is to confirm if sex hormones, but not chromosomes, may have impact on diastolic performance. As you can see here, the gonadectomy in males resulted in elevated E prime ratio, so somehow reflecting, let's say, worsening of the diastolic function. And in gonadectomy in females, lead to reduction of E prime ratio, EA ratio. So somehow we can conclude it's kind of improve state. And left ventricular ejection fraction was preserved. So somehow the model based on this looks like more or less adequate. Next, they came back to the model of heart failure with preserved ejection fraction, this two-hit model, but without any additional influences. And they measured mitochondrial DNA content, and consistently with previously shown results, mitochondrial DNA was lower in female hearts compared to male hearts, and also further decrease after heart failure with preserved ejection fraction made this decrease deeper. Also, basal state 3 and state 3U maximal respiration upon heart failure with preserved ejection fraction in females was reduced, as you can see, and mitochondria in females exhibited lower activity of complex 2, as you can see here, and complex 4 compared to males. So this demonstrates that mitochondria in females was impaired during the progression of heart failure with preserved ejection fraction. And to confirm this, because this data was made on isolated mitochondria, they performed more or less same analysis, but on isolated cardiomyocytes. And as you can see here, maximal respiration and basal respiration was significantly lower in cardiomyocyte. So next, they try to investigate the underlying mechanism of impaired diastolic performance in females. So they try to investigate somehow some molecular signatures different between
Can we ask a question, please?
They performed RNA-seq for hearts from these groups of CHO diet to heat model, males and females, and principal component analysis of different differentially expressed genes exhibited 4 main clusters, as you can see here, related to sex and to disease. The genes related to sex was sorry, and then
can we ask a question?
I can't hear you.
Okay.
I don't know why. Let me check one thing. Just a second. Right. Sorry. Go ahead.
Yeah, I think Rashad had a question, and I had a question also. Earlier, the point. Why did they focus on mitochondrial function in the first place? They must have had an idea about that. So what was the reasoning behind it?
You mean?
Do they say why they focus on mitochondria? Because there can be a lot of
yes. You mean this slide, right?
Yeah, exactly.
So this is really kind of weird, as I said, because they before they did some research on isopatrienol-induced cardiomyopathy, and they refer to this data, and somehow, as I told you, they didn't provide link and any reference to this research. That for me is a bit weird, and somehow they didn't. The main explanation, as far as I understand here, that after the making correlation of expression of genes with the EA ratio, they found that these genes enriched in mitochondrion, cycle Krebs, and mitochondrial inner membrane. So this is more or less as that could be explained. But honestly, I think, I don't know, they just did this previous study, and I don't know, by any reason, decide to focus on mitochondria. But formal reasons, as far as I understand, just because of this, because mitochondria.
They don't say much. They say that, as you said, many of the transcriptomic hits are on mitochondrial function. So they said, why don't we go deeper in our model?
Okay, thank you. And Rashad had another question, I think.
Yeah. I was just wondering. So, is all the data from the heart? So, did they look at only the heart mitochondrial DNA? Or?
They claim left ventricular mitochondria.
Left ventricular. They didn't look at any. So all the data you're showing is just from.
From heart.
From the heart.
Yes.
There was also one slide when they showed in the male mice, they show that E over A ratio after gonadectomy.
Okay.
Go next. Yes, here. So they showed for the females the E prime and the EA ratio. Are they the same? Do they give the same information? These two metrics?
As far as I understand, they reflect ventricular filling, but EA ratio is a ratio of blood velocity in early and late diastole. So E early and A late diastole, and E prime, E the same blood flow, as far as I understand, in the start of diastole, and E prime is the movement of the mitral
myocardium.
Okay, because they show here for the females 2 different metrics, but for the males they only show the E prime.
Absolutely, I agree, because for me it also was a bit strange. Yeah,
Okay.
and that's why also I have some questions to this paper. If you further, you also will see some parameters present in females, but not present in males, so not published in males. So for me, it's also kind of curious, let's say.
Yeah.
Right. Okay. Thank you.
Yeah, thank you very much for the questions. So
You are the PCA. Yeah, here.
yeah. So as I told you, so they revealed these 4 main clusters, which were related, enriched in genes related to the inflammation, fatty acid metabolism, for example, cholesterol efflux. But they didn't go further, and based on complicated bioinformatical analysis and so on, they chose one gene, one candidate gene called ACSL6. It's acyl-CoA synthase long chain family member 6. It's a gene that encoding mitochondrial protein involved in fatty acid metabolism. And somehow it's catalyzed formation of acyl-CoA from fatty acids. So they focused on this protein, on this gene, and found that in this hybrid mouse diversity panel of more than 100 strains, there was a decrease in expression level of this protein in females that is consistent with mitochondrial DNA content. Also, for this gene, they found that it was significantly decreased after heart failure with preserved ejection fraction, as you can see here, and also consistently with the data on mitochondrial DNA after gonadectomy in females, which demonstrate less mitochondrial DNA level. These genes after gonadectomy, this gene was downregulated, and in female upregulated. So, and also, by the way, I don't know why, but by the way, they also noted that fatty acid synthase gene and acetyl-CoA carboxylase 1 was also downregulated. So this supports the possibility that impaired fatty acid metabolism contributes to the increased susceptibility to heart failure in female mice. Next, also they found one single nucleotide polymorphism, which related to the increased expression of this gene as ACSL6, and this polymorphism, presence of this polymorphism GG, somehow negatively correlated with the EA ratio. And in other traits on this hybrid mouse diversity panel, they found that both in males and females, expression level of this gene negatively correlates with the E prime ratio, EA, and EA ratio, and some other traits. And they went further and choose 2 strains with a quite dramatic difference in expression of this gene. So one strain AG and another one KKHIG-J. So as you can see here in first, it's increased expression level of this protein, another one decreased, and this somehow negatively correlates with the E prime ratio and also negatively correlated with the glucose tolerance. So they propose that this protein ACSL6 could be important in the regulation of diastolic dysfunction. And to confirm this, to validate this,
can I ask something?
Yes, of course.
In the previous slide, so how do they determine these strains? Do they just check which strain has this difference based on sex, and then they?
Yes, I will. Sorry, I will go to the here. So this is the experimentally was the panel of these strains, and this classical C57BL6J, and here is this one strain, and this is another one strain arranged based on the ACSL6 expression.
And then they just why did they choose these two strains that had this difference, that's sex difference?
So they choose this to demonstrate then that the high expression of strains with high expression of ACSL6 demonstrate lower E prime ratio, and strain with the lower expression demonstrate opposite higher E prime ratio.
Right, because it seems to me that they would have checked all the different strains and checked just the difference between males and females, and then described the one that did have the sex difference.
Yeah, I think the idea is somehow to show that the causative, so it's causative for this exact gene, causative for
in these strains in particular.
Yes,
in these two.
Yeah.
Okay. Thank you.
As a sort of proof that a genetic background with different expression naturally, and this is sufficiently correlated with this diastolic, this filling parameters and this function parameters.
And honestly, this for me was kind of surprising that among different strains you can have. I mean, it's kind of logical, but I mean, for the researcher, they choose the proper model somehow. Again, very important.
But then I would imagine there would be a reasoning to choose these strains. Otherwise, it would be a replicable sort of difference in all of the different strains. That there is this difference in the sex difference in this specific gene in all of the different strains. But in this case, it's only two of them.
Yeah.
Well, it's just they're taking at the extreme.
Yeah.
Taking at the extreme of the panel. So we have the KKHIJ that is in the lowest expression naturally of this acetyl-CoA synthase. Pardon, acetyl-CoA synthase, and the AJ is the highest one, and naturally, and the BL6 is somehow in the middle.
Yeah,
and I think it puts. I don't know this database. I think that if the database is accurate, it puts quite elegantly that there is a crosstalk between a genetic background and sex that sustains that at least determines to some extent the expression of this protein. Then, but maybe later they have this proof that indeed it's correlated with FF markers. The next slide. This one.
So can I?
Yeah, go ahead. Sorry.
Yeah, no, thank you. So to validate the impact of these proteins, they on diastolic dysfunction, they overexpressed this ACSL6 in hearts of normal C57 BL6J mice using unassociated viruses. And then induced heart failure with preserved ejection fraction to test whether higher ACSL6 levels could ameliorate diastolic dysfunction. As you can see here, so here they demonstrate the expression level between control and overexpression. Here is, and authors also write about this kind of limitation of the study, that expression of this gene was also significantly increased in liver, adipose tissue, and muscles. But anyway, overexpression of these proteins somehow abolished the effects of two hits model. As you can see here, it's improved glucose tolerance, it's improved exercise performance, cholesterol levels was improved, and body weight and fat mass was also improved. And what the most important that for us, that overexpression of this gene led to decrease of EA ratio and E prime ratio and left ventricular mass with preserved left ventricular ejection fraction. So somehow they declare that this gene beneficial to the improvement of diastolic function, and consistent generally with this, the electron flow of complex 1, 2, and 4 was also increased and was also activated. Yeah. For me, for example, here is also saying about disadvantages. I also would like to see mitochondrial DNA content here, but I don't know why don't provide this. For me, it would be also curiously to see if it's affects on this gene effect on, but they didn't provide. But they somehow suggest that this protein improves mitochondrial function and helps to prevent diastolic dysfunction in favored preserved ejection fraction.
What's this
Sorry.
here?
TC total cholesterol.
Okay. And this what is
I think this area under curve.
which area?
For this glucose.
Oh, okay, for the glucose
for
tolerance.
GDT and glucose tolerance test.
Okay, it's not clear at all
Yeah,
from the text.
It's absolutely. So they use the same approach in females, and again, overexpression was less, but still present, increased in liver, muscle, and kidneys. And in general, again, overexpression on this protein abolished the effect of this two-hit model. For example, same glucose tolerance was improved, same cholesterol levels was decreased as well as free fatty acid levels. Running distance was decreased. And for example, here they provide expression level of natriuretic peptide precursor B, and they demonstrate that it's also decreased, but somehow in previous in males they didn't do this. But this means they suggest that somehow this is result of the improvement of metabolic state, general state, and same. Diastolic traits characterizing diastolic function was decreased, kind of this E-A ratio, E prime ratio, left ventricular mass. So somehow it has overexpression has beneficial effects, but it didn't affect any mitochondrial complex respiration, and authors said that this is due to some sex-biased mechanisms. They don't have a lot in discussion, honestly. They mostly describe this result observed, but generally in conclusion, based on their discussion section, we can suggest that, or they suggest that males tend to have higher levels of mitochondrial gene expression and also mitochondrial DNA content than females. Levels of both gene expression and mitochondrial DNA levels regulated by sexual hormones. Mitochondria contribute to diastolic properties, and this SL6 protects against diastolic and metabolic dysfunction in sex-biased manner. So, thank you.
Yeah. Very good.
Thank you, Andrei. Very good summary. There are a couple of questions in the discussion.
I have a question.
No, it was you. Okay, it was you both. Okay.
Can I ask a question?
Please.
I'm still a bit concerned, or I'm not sure about causality. I think I see an association between the observations, but is there a conclusive evidence that there is a causal relation between the mitochondrial function and the diastolic dysfunction? Or is it just an association?
It's, I think, very reasonable question, because for me, there is also, they provide more correlation that somehow this one trait correlate with another one trait and so on, but correlation doesn't mean real some causative relations. Yeah, so for me, no.
How can that be tested, actually, the causality?
Yeah, well, I mean, all in those lines. The link to calcium could be interesting to look at diastolic calcium and all of the proteins and ion channels that handle this.
Yes.
And then linking that to see if it correlates with these protective kind of mechanisms or things such as the changes in the mitochondria.
Yeah.
But yeah, that's a whole nother project. But
no, but I was thinking along the same lines, Jason. We know that the SGLT2 inhibitors have a beneficial effect because of their lowering effect of calcium. And as a consequence, they increase calcium in the mitochondria, which is protective, I would say. So there could be a causal relation between the phenomena if you involve the calcium concentration. What do you think, Guido?
It could be. I must say that the mitochondrial characterization is fairly basic, and it's based only on the amount of oxygen consumed. We have no clue on the amount of ATP produced. We have no clue on the type of preference in terms of substrates. I'm not surprised that there is a decrease of fatty acid synthetase because these animals are fed with an high fat diet content. So it's normal that it will revert to a more glucose-like phenotype. This is not anyway. It's not in any way impairing the study critically, in my opinion, because a bioenergetic switch is typical to many cardiovascular insults. I think that in assuming that the mitochondrial gene expression is normalized, which I suppose it is.
Yeah,
it's normalized to the relative number of amount of DNA, mitochondrial DNA, nuclear DNA. What I can retain from this paper is that okay, there is a higher expression of mitochondrial genes in male versus female. This is interesting. Again, if it doesn't depend on the mitochondrial mass, okay, I can live with it. Very interesting. I don't know if you mentioned this in your presentation. They did also this same analysis in human healthy and human failing hearts.
Yeah, they used previously published data of some open source data, so it's not their own data, but published before.
But are they in the presentation?
Yes.
Yeah. Okay.
Yeah.
Can you show them again, please?
Yes.
Yeah.
It's gonna be low because you are in presentation and you are in. So now it will jump 15 slides ahead. Okay. Here. Here. Yeah, you see.
Human horse. Yeah.
So this is in human.
Yeah.
So okay, this is more convincing a little bit. That when in general there is more mitochondrial production, probably synthesis in males versus females, and when the heart is failing, the same. So this is relatively interesting, and I'm quite convinced on the mitochondrial copy number inverse relationship with the specific with normal or degenerated heart function, because we might have a lot of mitochondria which are but they come from the same strain that reproduces because many of them are dysfunctional. So how to prove causality? This is very complicated, very complicated.
Yeah.
And this gene that they have selected, I'm sorry, it doesn't really convince me. What I am really surprised is that in female, the overexpression, it does nothing to the mitochondrial function. Nothing, like zero. This is something I would like to ponder upon. Especially because we are starting a study which is fairly similar, not in terms of diet, but at least the project that Andrei will work on, it's on ovariectomy of sheep to induce surgical menopause. And then I'm expecting many of these things, such as reduced mitochondrial copy number, reduced mitochondrial DNA, and some mitochondrial dysfunction. But which one? And now I'm not sure.
Okay, may I also have a question? And it's going to be very basic because I'm not a big expert in this field, and this was really like I was overwhelmed by the number of all the figures that was in the paper. So I will go with a very, very simple question. So I always thought that basically the mitochondria, like males get mitochondria from their mothers, right?
Indeed.
So basically the mitochondria, I kind of imagine that these mice were kind of sort of relatives, that they were like breeding them in the same facility. So I would guess that like the mitochondria are pretty similar in them. So all the differences we see are just caused by the effects, like the differences male versus female, are just caused by the effects of sex hormones, or is there something else that changes the functions of mitochondria? Because basically this paper shows quite big differences in mitochondria between males and females, but in the end, like the mitochondria come like from the, like they have like same mothers. Like imagine it's like a big family we are like studying, like big mouse family. So so do like sex specific hormones, or do sex hormones have such tremendous effect on mitochondria then?
Well, I will let Andrei, if you have an answer on this, go ahead. But Vladimir, I want to specify that only the mitochondrial DNA is heredited from the mother exclusively. So mitochondrial DNA encodes very little proteins of mitochondria. Most of mitochondria are encoded in the nucleus of cells. And there are around 26 proteins that are encoded in the mitochondrial DNA.
Okay.
Very little of them. Of course, the mitochondrial DNA is a bacterial DNA, so it has no way of base excision repair, double strand break, cannot do any of these things. So it's very much susceptible to, vulnerable to oxidative stress or to errors in duplication. This is why mitochondria are duplicated very quickly when is necessary, because they cannot repair their DNA. You have something on the sex hormone regulation?
No, I generally agree, but I just want to say also that sexual hormone somehow has a, as we can see, should have some impact, but general background, as they shown, for example, here on this slide, you can see this diversity in because this each point it's means different strain, and you see between the different strains the high diversity of mitochondrial DNA content. So somehow genes, really nuclear genes, affect at least the content of mitochondrial DNA and ability to.
I really hope these data are normalized, because it's not written there. I really hope that these are normalized. Anyway, this is one of the major obstacles in knowledge that we are finding in this project, is that we know that at least some levels of hormone association, hormone levels, is associated with cardiac hypertrophy, and we know that cardiac hypertrophy is associated with an increase of mitochondrial mass. And potentially a switch to a more fast and less oxidative dispendious use or production of ATP. To make it simple, every heart that is stressed for a short period of time will switch to glucose with respect to fatty acid, because glucose produces less oxidative stress. But this is only temporary. The heart cannot work on glucose for too long. Otherwise, it will try to. There will be glucose deposition inside the heart, and the heart will try to supplement the Krebs cycle with other energetic substrate, which are not good. Well, they keep the heart pumping, but in the long term, they're not good. And this is what causes, in the long term, this pathological remodeling that lead to systolic dysfunction, for example. Of course, for the systolic dysfunction, the reason might be a little bit more nuanced. Might involve much more calcium buffering, for example. But at this stage, I don't know. And I guess you don't as well. But we hope to see something in our sheep model in 6 months, and to be able to at least to give some general extent of what's going on.
Are you planning to include an SGLT2 inhibitor group?
We don't, but we have a progesterone replacement group.
Okay.
So to induce this sort of false pregnancy mode, which might be cardioprotective. We don't know how. We will see. Because estrogen, as probably all of you know, they are incredibly difficult to control in terms of side effects.
Yeah.
Progesterone is more controllable. Is there any other question? This is a very dense paper, and I don't agree with many things that they did, but especially this jumping from one model to the other. The isoproterenol model, I'm sorry, is not a model of FF, but okay. Actually, isoproterenol stimulates glucose utilization a lot. So, any other question? We are very much on time. No, stunned and philosophically pensive silence.
I have one detailed question about gene expression data. They are normalized to actin, I think.
For the West, yes.
So how does that work in hypertrophy? I think these cells are hypertrophic. If you take the wrong control, then everything will go down because actin will be more produced than the other proteins or the genes of other proteins.
So, it is an excellent question. I'm not sure. The bands they look very similar, the control bands. So I'm not sure. It might be monomeric actin that you see in Western blots. This is just an assumption.
You had data about gene expression, not so much Western blots.
Oh, right, sorry.
Maybe you can look at them.
Yeah. I was looking at
nuclear DNA.
I would suppose that. I'm not sure if of course, this is unlikely. Tubulin expression, I know that is changed a lot. GAPDH is changed a lot in condition, even atrial fibrillation. I'm not sure. I wouldn't have used actin. I wouldn't have used actin.
Here they claim, yeah.
Do they have a rationale for using actin?
No, I mean, no, but I mean, for gene expression, so they said like mitochondrial DNA content was normalized to nuclear DNA.
Yeah. Of course, like this is the way you do it.
But for this,
for the ACSL6, where they use actin as control. No. I guess the evidence. I mean, if we believe the gel, I think the evidence says that they are more or less the same.
Okay.
In terms of expression, we have to believe the gel.
Yeah.
I wouldn't have used a structural protein when there's suspicion of hypertrophy.
So what would you have used? GAPDH or?
Probably GAPDH if I were sure that there is no substrate switch. If there is no more glucose dysregulation. I would have even tried beta-actin just to see what's going on.
Okay.
It's weird. There is this paradox that I find in literature all the time. I must say that I don't do a lot of expression, and usually here we use GAPDH. Mainly I do Western blots, and I don't bother anymore with the control band. I use the total protein background.
Yeah.
This simplifies a lot. Of course, the total protein background, if you read around it, you have to be sure that there is not a lot of tryptophan in your lane, which is a pain. But in general, these conditions are met.
Okay.
Right.